Good morning, everybody. It's Dr. Nick Samadani. Welcome to Anatomy and Physiology 102. This is for sections 31, 70, and 71. Summer of 21, of course. In this course, we're going to do a fast-paced study and learning of various systems of the body. So we're going to jump right into the nervous system today, actually. Talk about nervous tissue in chapter 12. First thing I want to do is congratulate you on finishing 101 and passing 101. So that's great for all of you. And you all know what you have committed yourselves to, I hope. So it's very fast paced. In fact, your first exam on chapters 12 and 13 is coming up next Monday on the 14th. So it's very fast paced. So keep up on the reading. I don't put down the reading, but every chapter you are responsible for reading the chapter. And now, how deep you want to get into it is up to you. As far as your time commitment is concerned, and your background and this kind of language. But I tell you, you know this for sure by now that especially if you're going into nursing or medicine, this is vital, right? This is like your foundation for future learning. And the more you can dive into it deeply and understand the material as best you can, the better off you'll be, not right now, but in the future as well. And I really encourage you to purchase the textbook and read the textbook. Even if you just have the ebook, that's fine also. But do read the textbook, do read the ebook at least once through, so you know where everything is. Because someone told me that you don't have to know everything, but you have to know where to look everything up. So when, let's say, you're going into nursing, and in nursing school they assume that you already know your A and P, so they're going to dive right into the pathophysiology, thinking that you know the basic anatomy and physiology. So if you need to look things up, you need to go back to your textbook or ebook and be able to do that. I recommend a print textbook, because then you can carry it around with you in case you need to, if you don't have internet access, or if you lose your subscription or something, or you stop subscribing. So it's a good idea to have a solid print textbook that you can refer to later on. All the PowerPoints are based on the 15th edition. So if you have the 15th edition, it'll be easier for you. Although if you have earlier editions, that will suffice also. I don't assign homework right out of the textbook, but the material on the exams are based on the textbook and the lecture. Okay. So let me share my screen and go over the syllabus with you guys. Okay. So this is for 3170, and the same holds true for 3171. So they're both the same material. So best way to reach me is by email or Canvas mail, Canvas messaging. I answer within 24 to 48 hours, excluding weekends and holidays. So if you email me on a Friday, you might not hear back from me until Monday. Although I do check my email on the weekends as well, but I'm not going to commit myself to it. this is my physical office, but I won't be there. There are no office hours during the summertime, although if you want to meet with me on Zoom personally, or in a group, you can email me and we can set up a meeting, right? But there's no office hours during the summertime, unfortunately. Lecture hours are 9 to 10:30, or 10:20, sort of, Monday through Thursday. And the lectures will be recorded, and they'll be made available the same day, same afternoon. But I do recommend, if you have the time and the availability of time, to commit yourself to the live lecture so that you can ask questions, sort of get it out of the way in the morning, and then be concerned with other things. This is my biography. Some of you have had me for 101, so it's the same biography. Tells you a little bit about me, what I went through, some of my challenges in life, and what I face today, with other challenges as well. And then this is the course information here. So we'll be talking about the nervous system, the cardiovascular system, endocrine, lymphatics and immunity, and then respiratory, and finishing off with urinary systems. These are the course objectives that we'll try to accomplish. These are the course learning outcomes. It's a fully online course. Lectures are asynchronous, although I have live lectures. You're not required to attend the lectures. Required textbook is the 15th edition of Tortora, Principles of Anatomy and Physiology. You need to have access to Canvas, so obviously it's an internet-based course. Best search engine to use is Google Chrome. You can get the images much easier on APR, on McGraw-Hill Connect. And then for the Connect, McGraw-Hill Connect, this is the ISBN for the $40 one semester access, 180-day access card. And this is the one for 540-day access card. This is the one I recommend. If you can afford it, pay the extra $30, $31, and get the 540-day. That way you can access it in case you need to study for the TEAS test or the MCAT or different exams. Okay, this is really important. To register for Connect, you need to go to modules, click on the first lab assignment, and it will take you to a page where you can register for the Connect. This is the grading policy. The points are approximate, so don't hold me to them. It's approximately 1,000 points. Approximately 1,000 points. So during the summertime, it's really, really important that you guys follow the deadlines, the due dates, because it's really difficult. It would be really difficult for me if I had a bunch of students who missed assignments to go back into the computer and reassign them the assignments with exceptions and extensions. And it would be really difficult for you guys as well to catch up if you miss these assignments. So make sure you look at the due dates on the assignments, okay? It's your responsibility to drop the class if you're not going to attend. I'm not going to go and search to see who's submitting assignments, who's not submitting assignments, and to drop them on time and all that. It's up to you. I encourage you to read through the core rules of netiquette in case you're not familiar with them. Basically, it's like etiquette for the internet, right? Um, and to be basically to be courteous and polite and respectful of other people's opinions, and know that you're on the internet. You're not the only one on there. Everybody else is on there as well. So whatever you share is going to be seen by other people. Um, this is something I want to read to you, the equity statement as well as the academic honesty, because they're really important, especially um, when you're dealing with nursing and medicine and dealing with people of various backgrounds, right? As a nurse, as a doctor, you cannot discriminate between different people based on your experiences with them, right? That's why doctors take the Hippocratic oath that above all do not do no harm. So you cannot say, oh, this person is from this country or this religion or this background or whatever. I'm not going to treat them as well as I would do my own kind, which is really ludicrous, because by now you probably all know that we're all different. Even though we appear to be similar, we're very different anatomically, right? Um, so we're different. We're different anyway, not just in um, obvious things such as skin color or uh, whatever facial expressions or whatever, um, but we're different anatomically. So it's a given that we're different. That's why they say, sometimes you have to put yourself in other people's shoes in order to understand what they're going through, because we're all different. Okay, so I believe that diversity in the classroom is what fuels fruitful discussions and learning, which brings me to this topic of discussions. I will post discussions and encourage you to follow up on them so you don't get burned out on this summer semester, because it can get pretty hectic trying to learn all this material. So if you form a discussion group online on Canvas and you converse with each other that way, that's another form of learning that I really encourage. I also believe that there can be unity in diversity. In Eastern philosophy, there is the concept of many in body, one in mind. We can be many in body, but we all want to succeed in the course, so we're all one in mind. So I encourage you to help each other, not on the exams, but on during your study time, right? To get on Zoom, to get on the discussion boards, to get on your own phone list or message list or whatever, and keep it interesting. Keep it interesting and interact with people. Um, Anatomy is something that's learned better in groups, because then you can bounce ideas off each other and you can pronounce things, um, and since we don't have the advantage of being in the classroom, I really encourage you to get on the discussion boards and do that for the summer. As far as academic honesty is concerned, I'll read this as well. Many of you are planning on entering fields such as nursing and medicine, which demand rigorous honesty. You will be in charge of people's lives, and therefore to not be honest in your studies would be like a double-edged sword. Double-edged sword because it would kill the patient and it would destroy yourself as well, your career, background, your reputation, and so forth. Practice honesty now, and as we were in medical school, you're on the honor system, which means that I trust each of you to do academically honest work. Each of you is also responsible for not sharing exam information with other students. I encourage each of you to set up discussion groups and have Zoom meetings to go over the material covered in lecture and the textbook. But during exam time, please accept responsibility for what you know and turn in honest work. Okay, so that's really important. So I'm not going to set up cameras or things like that to proctor the exams or anything. You're on your own, but do take that academic honesty into consideration. Academic support, first off, the library and the Learning Resource Center are going to be open during the summertime. So do take advantage of those, right? We have Charles here. Hello, Charles. Charles is one of our IAs, instructional aides. So um, he will be available to you guys as well in case you have questions. He's a long-term veteran, not just a veteran of the armed forces, but veteran of anatomy and physiology. So he knows a lot. So you can uh, use his knowledge as well. Okay. If you have questions related to technology, this is Academic Technology Services. You can click on this link. It'll take you there. If you have any technology questions. This is the Student Services Online Hub, and it has the disability services in it. It used to be called DSPS. Now it's called ASC, right? Accommodation Service Center. Okay, so if you think you have a disability, it's a good idea to get tested so that you can get accommodations. And if you have a certified disability with the ASC, you need to contact them in order to get the accommodations that you need, right? Extended test time and so forth. So that's it for the syllabus. Does anybody have any questions about the syllabus? Anything you want me to go over?
Can you hear me?
Yes, go ahead.
So I wanted just to confirm that all that we need for this course would be the 15th edition AMP book and then the $40 virtual lab, right?
Yes, yes,
Okay, thank you, Professor.
yes, that's all you need. Okay. So let me jump to the schedule. So the schedule is pretty self-explanatory. It has the dates, the lectures, chapters of the lecture, the topic we'll be covering in the lecture, and the lab activity for that day. So for example, today we're going to jump into the nervous tissue in chapter 12. That's the first lecture. And then for lab, you're going to do the virtual lab for lab safety and hand washing in Connect. Okay, and I'll show you how to access that right now. Okay, so this is my home page. Again, the required textbook is written right here. This is without Wiley Plus. You don't have to purchase Wiley Plus. And then this is the electronic platform from McGraw-Hill's Connect. Either the $40 one or the $71 one. Either one will work. Okay, and this is the meeting ID. It's always going to be the same unless I change it, and I will let you know, of course. But I plan to keep it the same. So this is the meeting ID. And then I want to go to the modules. Click on the module. If you haven't seen these in previous classes, it's a good idea to go through them. For example, the netiquette is over here, and it talks about the core rules of netiquette. Remember the human, adhere to the same standards of behavior that you would practice in person, You're not the center of cyberspace. Everybody else can see your post and everything. Make yourself look good, share expert knowledge. Don't just blurt out shoot from the hip, just give an answer just because. Make sure it's research. Be forgiving of other people's mistakes. Never be arrogant or self-righteous. So go through these if you haven't seen them before.
Excuse me, Professor.
Yes.
Where can we find the recordings of our Zoom lectures?
I'm sorry, I didn't catch that.
Where can we find the recordings of our Zoom lectures?
I will post them each afternoon. I will post them in the announcement, and then I will put them in the module as well. So it'll be a link basically to the lecture, to the Zoom lecture. And I will share the link with you guys that same afternoon. That's why I say if you can make it to the morning lecture, you'll be much better off, because then you don't have to wait till the afternoon to get the lecture, to get the recording. Okay, so let's jump to week 1. So week 1 has the lecture on the left and the laboratory on the right. The lecture has chapter 12 lecture PowerPoint, which you can open up. It's in PDF format, but it's the same PowerPoint that I use for my lectures. This is the chapter 13 PowerPoint. So we'll be covering 12 and 13 this week. This is the study guide for the chapter 12-13 exam, which is coming up next Monday, the 14th. So that's really quick. The study guide points you to the right direction, so make sure that you know the study guide well. Okay, I go over the study guide in my lectures, so make sure you do the study guide. Maybe take it to the LRC or the Open Lab, which we really don't have, but you can share it in a discussion group or form your Zoom group or whatever. You can go to the LRC tutor or net tutor. So there's many, and you can also set up a Zoom meeting with me and go over the study guide if you have questions about some of the concepts. Okay, as far as the lab is concerned, so this week we're concerned with chapter 12 and 13. And as far as the lab is concerned, the virtual lab, this one is virtual lab safety. And I see one student has already submitted, so that's good. And that's how you access it. And then if I can get looking for student view. Okay, so that's how you access it. I'm gonna go back to modules. And hand washing too, you need to do today. And then you have 2 weeks to study the nervous system in the practice atlas and in the APR. So take note of those 2 things. Practice atlas is different than APR. APR is based on cadaver pictures. These are real life cadaver pictures that are very useful for learning realistically, right? So let me go to the practice atlas. So what you do is go down here on the left navigation pane over here. McGraw-Hill Connect, right? Go to Connect. Go to my Connect section. And then I want to get student view so you guys can see what I'm doing. So in the student view, you have Practice Atlas for Anatomy and Physiology, which is right over here on the left side. You click on that. These are abdominal organs. We need to go to the nervous system. So it has study and practice, right? Study and practice. So first you're going to study the nervous system. Let's say we're going to look at the brain model. Okay. And it has like sort of interactive features that you can use. So you click on the cerebrum and the whole thing lights up, or the frontal lobes of the brain, or the temporal lobes, or the occipital lobes. This is the cerebellum, the little brain, and this is the brainstem. Right? Go down here, see where the olfactory bulbs are, the olfactory tract, the optic nerve, optic chiasm, and so forth. So it has interactive ways of studying the material and learning the material, right? So I'm not going to be going over all of this with you. It's your responsibility to go and learn all the material within the nervous system in the next 2 weeks, okay? And I've given you a code for APR that I share with you every week. So APR is down here. This is Anatomy and Physiology Revealed. This is based on the cadaver, basically. So they're based on cadaver pictures. You go up here on the upper left to the green box over here, green square, where it says My Course Content. Click on that. And then this is the code that I've already have from before, but this is the code that I gave you in your module, okay? So you're gonna go over here and add delete list. I'm gonna add list, okay? And you're gonna go on down here, you're gonna say add list, and you're gonna enter the code here, and you're gonna press OK. So let me see. It says duplicate code. So that means I already have it. So when you guys enter it for the first time, you're going to enter the code and you're going to click on OK, and it'll take you to the nervous system list. Okay, these are the list of structures that you need to know. So once you get the list, then you go to the module and you select number 7, nervous system module. And then all these things come up here on the left side. These are all in alphabetical order, okay? So don't study them in alphabetical order. Study them by topic. So I click over here in topic, and it'll give me the spinal cord, the brain, and the cranial nerves, as well as the special senses. So you're going to be studying the whole complete thing within the next 3 weeks, really, 2 to 3 weeks. Okay. So let's go to some of these here. Let's go to the brain, the coronal plane. The coronal, remember from 101 that it's basically dividing the body into anterior and posterior portions. So here's the entire brain that's lining up. This is the brain stem. Right? And we have the cerebrum, for example, which is the main portion of the brain. The frontal lobe, so on and so forth. So when you study this material, you can get more information on the particular structure that you're studying by clicking on I for information, structure information. So I click on frontal lobe. It gives me an location, description, and function, right? What it does, how it's put together, and where it's located. Okay, so if we want to study deeply, you can, and you can click on this to pronounce it.
Frontal lobe.
Frontal lobe. See it in different views. Okay, so that's how you're going to study the nervous system. You're going to read the textbook, you're going to listen to the lectures, hopefully, and you're going to study the APR and study atlas.
Excuse me.
Yes.
Are we gonna have, besides the exam, we're gonna have a separate quiz on the nervous system, like with the pictures to identify the parts, and this is what you are showing to study.
Right, exactly. The nervous system exam, there's a comprehensive nervous system exam that you're going to have, and that's based on all the APR, all the anatomy physiology revealed pictures.
And that's that next Monday? Where that?
No, next Monday is just chapter 12 and 13. So let me bring up the syllabus again and show you. Bring up the schedule. Okay. So you have a brain anatomy quiz due in Connect on June 16th. That's next Wednesday. And you have a comprehensive nervous system exam in Connect June 23rd. Okay, so that's based on the McGraw-Hill Connect APR. Okay. Okay. So again, this is the practice atlas here, and this is the anatomy and physiology revealed that you're gonna use. So, okay, any other questions on how to access information there in McGraw-Hill Connect? Okay, so if there's no other questions about accessing the material, I will start the lecture on chapter 12. So we have about 40 minutes of time to talk about the first part of this chapter. Chapter 12 is on nervous tissue. So we're going to talk about different types of cells that are found in nervous tissue, such as neurons and glial cells, support cells. We're going to talk about how the nervous system is structured, how it keeps conditions within limits, and maintains health and homeostasis. So the nervous system, in addition to the endocrine system, which is controls the body with hormones, um, both of these systems are regulators of the body, right? So they regulate specific metabolic processes or uh, homeostatic processes that their function and their purpose is to keep the body within homeostasis, right? Which means health, keeping the body in the same condition, homeostasis. So the nervous system is divided up into the central nervous system and the peripheral nervous system. So the central nervous system is over here in the yellow rectangle here. It consists of the brain and the spinal cord. Okay, the spinal cord and the brain are in the central nervous system. The peripheral nervous system consists of the cranial nerves. Cranial nerves, even though they're called cranial, don't think that they're part of the CNS, they're part of the PNS, or peripheral nervous system. So cranial nerves, spinal nerves that come from the spinal cord, enteric plexuses in the small intestine that control movement of the intestines and so forth, and sensory receptors in the skin that are going to pick up stimulation or changes in the environment and send that information to the central nervous system, namely the spinal cord and eventually the brain. Okay, so this is really important to understand and to know. And then this slide is very important to spend some time on and really understand it. The central nervous system here is designated in the yellow rectangle here. So central nervous system. And the peripheral nervous system is in the purple rectangle here. So all of this is peripheral. And all this is central. Now the peripheral communicates with the central, and these are not like separate pieces of the same system. These are all part of the same system. It's just that human beings like to compartmentalize things and organize things and so forth to understand things better, but they're part of the same system. They're always communicating with each other and interacting with each other. So, for example, the peripheral nervous system picks up sensations such as touch or pressure or vibration sense or something through the somatic senses and communicates with the central nervous system that way, right? Somebody touches us lightly on the skin, and that gets translated into a message or a signal that gets sent to the central nervous system, the spinal cord. And maybe we have a reflex and we like deflect our hand to get away from them, not like as a reflex. So the somatic senses are involved with the detection of change in the environment. And the spatial senses have to do with eyesight and hearing and taste and equilibrium and balance and so forth, and smell. So those are the spatial senses. They're also going to communicate with the central nervous system via sensory input. So we have a sensory division of the peripheral, and we have a motor division of the peripheral. Now, the motor division is divided up into the somatic nervous system and the autonomic nervous system, right? The somatic nervous system is going to control skeletal muscle. It's going to innervate skeletal muscle and cause movement, right? Voluntary movement. The autonomic nervous system is divided up into the sympathetic, the parasympathetic, and the enteric nervous systems, right? So the sympathetic and parasympathetic, they do different things, and we're going to talk about them in a later chapter in detail. But they control smooth muscle, cardiac muscle, and glands. So remember that the autonomic is really part of the motor division, right? That sends signals to smooth muscle, cardiac muscle, and glands, and makes them contract or makes them secrete, right? The glands, we mean like sweat glands and salivary glands. These are controlled by the autonomic nervous system. And the autonomic, you can think of it as sort of automatic, right? Autonomic is automatic because we don't think about all these processes going on that are governed by the autonomic nervous system. They sort of occur automatically. And then the enteric nervous system is going to innervate smooth muscle and glands of the GI tract, right? The GI tract. And that's part of the autonomic as well. Any questions on this slide? No questions? Okay. So the functions of the nervous system are to pick up sensations, to deliver motor commands, and to integrate information, to analyze information, store some aspects of it, and make decisions regarding appropriate behaviors. So the sensory and the motor and the integrative all work together, right? The sensory picks up a sensation, the integrative processes that information, and then sends out a command, right, to the motor, for example. So they're all involved together, right? But these are some functions of the nervous system. As far as the histology of the nervous system is concerned, first we want to talk about neurons. So nervous tissue is basically composed of neurons and glial cells. We have more glial cells, about 6 times more glial cells than we have neurons. And neurons are electrically excitable, whereas glial cells are not. Neurons are designed to conduct electricity or conduct action potentials, right? We call nerve conduction action potential along the axon. We'll talk about action potential later on, but for now, know that neurons are electrically excitable. They have to be in order to communicate with other neurons. And this is an example of a multipolar neuron. Example of a multipolar neuron. This is the same neuron that you studied in histology in 101. It's multipolar because it has multiple dendrites. These are called dendrites. Here, dendrites written right here. And these are responsible for picking up sensation or picking up impulses and sending them to the soma or the cell body right here. The soma or the cell body houses the nucleus as well as the cytoplasm. And within the cytoplasm are structures called nissl bodies, which are aggregates of endoplasmic reticulum, because the neuron has to make a lot of protein, axoplasm, right? And it makes the protein in the nissl substance, in the endoplasmic reticulum. Now, there is a axon hillock right at the junction of the soma or cell body and the axon. This is the axon here, written right here. It's the axon. So the axon hillock is where action potentials initiate from in the axon. And that's the initial segment, is the initial segment of the axon. Okay? So action potentials or nerve impulses are going to travel along the axon, and they're going to reach the axon terminal here and end up at the synaptic end bulb. Synaptic end bulb because it makes a synapse, makes a connection to another neuron perhaps, or an effector cell. Could be a skeletal muscle cell, for example. Okay. Now, this particular axon is myelinated, and the myelination is not continuous. It has interruptions here, 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 here. For example, this one over here that's enlarged for better viewing. This is the node of Ranvier. Node of Ranvier, which is between the two internodes, right? So this is an internode, node of Ranvier. Why are nodes of Ranvier important? Because they have the highest concentrations of sodium ion channels. Now remember your chemistry now, because it's going to come and haunt you in a way, hopefully not. But the nodes of Ranvier are where you have the highest concentration of sodium channels that can open up and let sodium in, and that's what causes the action potential, as we'll see later on when we talk about the action potential. But the action potential in a myelinated axon is going to jump from node to node, and therefore is going to travel a lot faster. And we'll get to that in a later slide. Now, structural classification of neurons goes like this. We have the multipolar neuron, which is the most common neuron in the body. Most neurons are interneurons, and most interneurons are multipolar. Okay? So the multipolar neuron has multiple dendrites, a cell body, and a single axon that ends at the axon terminal and the synaptic end ball. A bipolar neuron, now let's say, I want to mention this, that the multipolar neuron is the most common neuron in the body. All the motor neurons are multipolar neurons. Okay, all the interneurons are multipolar neurons. Most interneurons are multipolar neurons. And then the yes question. No. Okay, so the bipolar neuron has a single dendrite and a single axon. Now it has dendritic spines, but they all culminate in a single dendrite. That's why it's called bipolar, because it has two poles, this one here and this one here. There's a cell body in the middle between the dendrite and the axon, and it has axon terminals. So bipolar neurons are found in the retina of the eye and also in the olfactory epithelium. The olfactory nerves are really bipolar neurons. And then unipolar neuron, also called a pseudo-unipolar neuron, because it used to be a bipolar neuron until its cell body got squeezed to the side with just one peripheral process going to the dendrites and a central process going to the axon terminals. So pseudo-unipolar neurons, they have their cell bodies located in the dorsal root ganglion of the vertebral column or the spinal cord. We'll see that later on. But sensations from the periphery, such as the skin, are going to come in through the dendrites. They're going to come over here to the peripheral process. They're going to get processed and sent a signal to the central nervous system, to the spinal cord, for instance, right? That's how pseudo-unipolar neurons work. So we have multipolar neurons, the most common in the body, bipolar neurons, and unipolar neurons, which are most of the sensory. Okay, multipolar neurons. A couple of examples of those are the pyramidal cell in the brain, the cerebrum, and the Purkinje cell in the cerebellum. The cerebellum is in the back of the head. It's called the little brain by definition. And this is the pyramidal cell. It's located in the cerebrum. So we'll see that over here. This is the pyramidal cell with its single long axon and multiple dendrites. Here. See the dendrites barely make it out here. So these are pyramidal cells of the cerebrum. And then these are Purkinje cells of the cerebellum. The cerebellum has a molecular layer, which is hypocellular, has less cells in it, less dense. and a granular layer that's more dense with cells in it. It's granular, right? It has a lot of cells. And the Purkinje cells lie in the Purkinje cell layer right here. And they can have as many as 10,000 dendrites. They can have 10,000 dendrites bringing in information for processing. The cell body is going to process that information and send a command to the axon. Now, why is the cerebellum designed like that? Because the cerebellum really receives a lot of information from different sources, especially the proprioceptors, right? These are spatial receptors on our joints, on our bones and muscles and so forth, that are going to tell the cerebellum about our body position and our body condition, right? Whether we can do a backflip or not, or whether we can do a break dance or not. These are all information that come in to the cerebellum. Cerebellum has to integrate that information and send out a command to the cerebrum and to the rest of the body, basically saying that, yes, it's feasible to do that movement, or no, it's dangerous to do that movement, to not do it. That's just as an example of what the cerebellum does with these Purkinje cells. Neurons can also be classified functionally, whether they are sensory or afferent neurons, or motor or efferent neurons, or interneurons or association neurons. So sensory neurons just bring in sensory information. Afferent means toward, so toward the central nervous system. Motor or efferent goes away from the central nervous system to the periphery. And interneurons are in between the sensory and the motor. You can think of it that way. So again, most interneurons are multipolar. All motor neurons are multipolar, and sensory neurons are mostly unipolar, although some are bipolar as well. So this is an example of a sensory neuron in the peripheral nervous system, in the purple, bringing in impulses to the central nervous system, where it innervates the dendrites of this interneuron, usually multipolar. It's going to send nerve impulses to a motor neuron, also usually multipolar, and that sends nerve impulses to the effectors, muscles, glands, and so forth, right? So very important concepts to get here, that how do we get information to the central nervous system, and how do we get information out of the central nervous system, right? Okay, so that's the neurons. Okay, the neurons again are electrically excitable cells of the nervous system, right? Electrically excitable cells. That means they can conduct action potentials. Now, neuroglia are not electrically excitable cells. They make up about half the volume of the nervous system. So most of the nervous system is made up of neuroglia. They can multiply and divide, unlike neurons. Neurons, once they die, they don't come back. That's why someone who gets a stroke, depending on the severity of the stroke, if it kills the cells, they're not going to come back. But if it damages the cells, they could repair and regenerate, right? There's 6 kinds total of these neuroglia. 4 of them are in the CNS, central nervous system. 2 are in the PNS, or peripheral nervous system. The 4 that are in the CNS are the astrocytes, which look like this, look like little spiders. These are the astrocytes. And one of the main things they do, which is a question on your first exam, is that they form the blood-brain barrier, right? So they have these protoplasmic feet, little feet, that wrap around this capillary here. This is a capillary. They wrap around the capillaries, as you can see, right here, right here, right here. They wrap around the capillaries and they form the blood-brain barrier. Blood-brain barrier is very important to the brain because the brain, first of all, the color tells you that it's not very vascular, right? It doesn't have a lot of blood coming in because it's like gray or tan colored or yellow colored in the fatty areas, in the interior sections and so forth. But it's not like the color of muscle, right? It's not the color of heart muscle or skeletal muscle or anything like that. And that's because the brain doesn't need to know about all the fluctuations in sugar and sodium and potassium and chloride and all these things that are happening in the blood. The brain doesn't want to know about it, except on certain parts of the brain that are designed for picking up information from the blood. And those are the paraventricular organs that we're going to talk about later on in chapter 14. Okay, but these are the astrocytes. Another thing that they do is that they form glial scars. So if there's an infection in the brain, they form a scar tissue around it, make it sort of like an abscess, and that's why it's hard to treat those brain infections. These are the oligodendrocytes. The oligodendrocytes are another kind of glial cell that are responsible for making myelin, rapid myelin around central nervous system axons, right? Central nervous system neurons and their axons. So they're going to wrap myelin around multiple different CNS neurons, right? Oligodendrocytes. Then we have the ependymal cells, which are part of the choroid plexus. So remember this term. It's going to come back to get you. Choroid plexus. The choroid plexus is made up of ependymal cells and capillaries, right? We have ependymal cells and capillaries. And the ependymal cells are responsible for making cerebrospinal fluid. Cerebrospinal fluid is the fluid that comes out of the spine when you do a spinal tap. And it wraps around the whole brain and the spinal cord in order to cushion it, protect it, and nourish it. Okay? So ependymal cells are responsible for making cerebrospinal fluid. And they're located in the ventricles, which are the cavities of the brain. Ventricles of the brain. Okay, next one is the microglia. Microglia are like the macrophages of the brain. Right? They're derived from monocytes. They travel to the brain and they become microglia. Microglia are responsible for vacuuming all the debris and waste material and degraded organelles and so forth in the brain, right? So the macrophages of the brain are going to phagocytize bacteria and debris and so on and so forth. So they're like the vacuum cleaners. So we talked about microglia, ependymal cells, oligodendrocytes, and astrocytes. So make sure you read about these in the text and know what they do. The neuroglia of the PNS, the peripheral nervous system, is made up of the Schwann cell, like here and here, and the satellite cell. So a Schwann cell is going to wrap phospholipid membrane around this axon, right? A single axon, and it's going to leave the node of Ranvier alone. So it's not going to myelinate the node of Ranvier. Remember, node of Ranvier is where you have the highest concentration of sodium channels. Sodium channels. And then with a satellite cell is going to be present in the ganglion. Okay, and ganglion is an important word to know, so you should know it right off the bat. Ganglion is a collection of cell bodies of neurons in the peripheral nervous system. The equivalent of a ganglion within the central nervous system is called a nucleus, not to be mistaken with the nucleus of a cell. This is a nucleus that has the cell bodies of the neurons, right? That's called a nucleus in the central nervous system, and the collection of cell bodies outside of the central nervous system in the periphery is called ganglion. And within the ganglion are satellite cells that are going to nourish and support the neuronal cell bodies. Okay, those are satellite cells. So these are electron micrographs of a nerve with a myelin sheath around it. So this is myelinated. This is myelinated. And then these are unmyelinated nerves, unmyelinated axons. Okay. Gray matter versus white matter in the central nervous system, the brain and the spinal cord. So in the brain, you cut the brain the coronal way, this way, right? You're going to see gray matter on the outside. So this is called the cerebral cortex, right? Cerebral cortex on the outside. And you're going to have white matter on the inside, which is going to be white, yellow, tan, glistening. Okay. So within the white matter, there can be some gray matter as well, such as the basal nuclei, which we'll get to in chapter 14. But most of the interior part of the brain is going to be white matter, right? It's not really white, it's really yellow matter, but they call it white matter. So this is a real actual brain, frontal section of the brain. And it shows you the gray matter within the white matter. Shows you the couple of ventricles here. These are the two lateral ventricles. Cavities in the brain that house choroid plexus, right? This is the third ventricle here. That's surrounded by the thalamus. And so forth. So we'll get into the structures in chapter 14. So that's the essential makeup of the brain in a concise way, very concise way. The transverse section of the spinal cord shows that the gray matter is actually inside and the white matter is on the outside. So opposite in the spinal cord, right? In the brain, the gray matter is on the outside, white matter is on the inside. Opposite is true in the spinal cord. Gray matter is in the inside, white matter is on the outside. What does the gray matter have in the spinal cord? It has mostly neurons. Okay, the ventral horn here has the motor neurons. The dorsal horn here has the sensor neurons coming in, and the lateral horn in specific parts of the spinal cord has the autonomic or the sympathetic. And then the white matter is basically made up of these elevators, you can think of them like that go up and down and they relay information to and from the brain, right? So the dorsal column, the lateral white column, and the anterior white column. Okay, so that's them in a nutshell. Okay. Any questions you guys have?
I don't have a question.
Okay, Lisa, thank you. Okay, so I'm not going to start a new concept until tomorrow. You have a lot to do today in terms of getting your proper literature and the Connect platform, electronic platform. You have some reading to do. So I encourage you to start reading chapter 12 if you haven't started already. And go over that. And once you get the Connect, start practicing on the Connect, doing the virtual labs and so forth for today. So please, please don't fall behind. It's summer school, goes super fast, which is the good part, right? It goes super fast, so you get it done early. Okay, any other questions or concerns, anything? Let me see the chat real quick.
Professor,
Satellite cells, satellite cells, there's a question in the chat. Satellite cells only present in unipolar neurons? No. Satellite cells are present in ganglia, peripheral ganglia. That's where the ganglion of the unipolar neurons is called the dorsal root ganglion. And within the ganglion are the satellite cells. Okay, this is something that Ruth brought up. Is that clear, Ruth?
Not quite. So in the
Okay.
ganglia, there are only unipolar neurons in the ganglia? Is that correct?
Depends on the ganglion, right?
Okay.
So we have a dorsal root ganglion, that's right next to the spinal cord, right? That houses the cell bodies of the unipolar neurons, the
Right.
cell bodies,
Right.
right? Now, within that ganglion are satellite cells that are going to support those cell bodies and nourish them. And
Okay,
satellite
so-
cells are also present in sympathetic ganglia, which we'll get into later on. But they're not just present in dorsal root ganglia, they're present also in sympathetic ganglia.
So I guess my question is more, are there satellite cells around multipolar neurons?
Around multipolar neurons, yes, in the sympathetic, they are multipolar neurons.
Okay.
Yes, in the dorsal root ganglion, you're right, they're unipolar neurons. Good question, very good question. Marcelo, did you have a question?
No, I guess I suppose we won't be reviewing I noticed that the questions for the chapter for the study guide are not in order, but I guess that's the point of it, right? For you to read the chapter.
Yeah. Yeah, but I will go over them.
Oh, okay,
I will
great.
go with the study guide on Thursday.
Thank you.
You're welcome. Anybody else has anything?
Is there a time limit for when the assignments are due?
The exams are due the same day. So make sure you pay attention to the exam due dates. The Connect exams and the lecture exams are on a specific day. So you have to take it on that day. Other assignments you can have a week on. For example, the lab assignments. I typically give you a week to do those.
Okay, thank you.
Okay.
So the lecture exams are not gonna take place during our lecture. You're gonna launch them, and we have to take them whenever we want during the day. Is it true?
Right, right. So on lecture exam days, we won't have lecture so that you have time to complete the exam. Okay, so the way the schedule is set up is that you don't have lecture on exam days. You see that?
Yeah, I saw that in the syllabus. Yeah.
Yeah, okay. So no lecture next Monday, only the exam, for example, yeah. Okay, anybody else has anything?